First, let me just give you sort of an overview, to kind of set the stage for the talk. As was mentioned, I'm from the Pacific Islands Fish and Wildlife Office in Honolulu, Hawaii, and we're part of Ecological Services. So our mandate is to administer the Endangered Species Act in the Pacific Islands. And that actually covers a very broad area. From the Hawaiian Islands down to American Samoa, and then to the Mariana Islands in the west. And this also includes quite a few listed and proposed species. Currently there's 571. The vast majority of them are in the Hawaiian Islands, and of the species that are in the Hawaiian Islands, the vast majority of them are listed plants. So as you can imagine, with such a broad area and quite a few listed species, we actually have, it can be very challenging to do conservation planning for these species. And what I'm going to be talking about today is our efforts to identify conservation habitat for these listed species. So the traditional approach, or the approach we've typically taken, is we've collected a bunch of data on the species locations, information about habitat, administrative units, things like that. Gotten together all our species experts and the biologists, and they've assessed this information, and they basically delineated these units by hand on a map or using GIS. And this approach works really well, but as you can imagine, it can be very time consuming, especially when you have quite a few species. And also, it's very hard to update. You know, when you have to get everyone together and then you have new information, it's very hard to update this process. And it's intended to be an iterative process. It's supposed to be changing as you get new information about each species. And then when you start adding in things like climate change, it becomes even more complex. And so what we wanted to try to do is find tools that will assist us with this process. And this is what the talk is about today. Specifically, I'll be talking about species distribution modeling or habitat suitability models, and then our efforts to rank and prioritize areas for these species. And so I'm going to be talking about the current climate, but the same methods could be applied to future climate under climate change as well. So before I get into the details of it, I first want to introduce our example species, Cyanea kunthiana. It's an endangered plant on the island of Maui. And this is the species I'll be using to kind of go through the process and explain what we're doing. It'll be our example species. So you'll see it popping up again and again. Okay, this is the methods overview. We're talking about three different things. One of the main things we want to do is, because we're talking about listed species, we have basically conservation criteria that we want to try to apply. The goal is to take the species off the endangered species list, so we want to basically delist the species. So those are our conservation criteria. And they're typically how many populations and how many individuals. And then the second part is we want to actually be able to determine where is the optimal area for these species on the landscape. And here we'll be using habitat suitability models. And then finally, I'll be talking about building and ranking those populations we have identified above on the actual landscape. So here's the population criteria. In general, these are listed within our recovery plans, if you're familiar with the recovery plans. They usually have some criteria to determine when you can delist the species. In the Pacific, we actually have a recovery committee that's in charge of all the different plants, and they've come up with criteria that apply to all the different plants in the Pacific. And the criteria for the species that I'm talking about today are 10 populations of 400 breeding individuals, and then ideally each of those populations will be at least 1 kilometer apart. So that's what we're shooting for. That's what we want to try to do. Now, in order to actually figure out how we apply that to the landscape, we actually have to know, well, how much space does 400 individuals take? And so that's where density comes in. And in an ideal sense, in an ideal situation, we will have done a whole series of surveys, preferably before the species was listed, where we can actually have a good idea of what the actual density is of the species prior to all the threats. Unfortunately, since we're talking about threatened and endangered species, this isn't always possible. So we have to come up with alternate methods to do that. And so in Hawaii, that's also the case. So we've been using this, what we call a pseudo-survey approach. Basically, we're using data on known locations for this species, estimating areas where people have searched for plants, and then deriving a density estimate from that. So it's a proxy, it's an initial value we're using that ideally we'll fine-tune and improve as we go on in the process. And so the value for this species is that density up there. So once we have the conservation criteria and the density, now we need to basically figure out where in the landscape we want these populations. And so here we're using habitat suitability models or species distribution models, and I'm sure probably many of you are familiar with these. They're fairly common now. And I'm not going to go into detail about how we do them and all that sort of thing. I'm just going to kind of talk about it briefly and talk about some details about how we do it. So the general concept is we have a whole bunch of locations for our plant, and those are indicated in the blue dots on this map here. And basically what we want to do is figure out, well, what's the relationship between where the species is and various other variables. Here we're looking at climate variables. And the idea is to use this information to develop a model about where the species might be. And there's multiple ways we could do that, all sorts of different modeling algorithms. But the idea is you develop the model, and once you're satisfied with the model, you project it onto the landscape, and it gives you a distribution or a suitability model across the landscape. So this is kind of some details about that process. I'm not going to go into it here. If you're familiar with species distribution modeling, there's some information here you may be interested in. But I do want to point out a couple of different things. One of them is there's no single model that works for every species, so we have to run multiple models, and that's part of the process of what we're doing. I've listed a few of them here. The other thing is that we're actually doing all this within R, the statistical program R. The main reason is partly because that program can run those different models, but also because it's a programming language. We're actually able to write a program where we can actually update and iterate through this process so that we can actually automate it to make it easier. So if we have new data and new information coming in, it'll be easier and faster for us to update this information. And it can be applied across multiple species. So we're hoping that that speeds up the process. The other thing I want to mention is all these models have to be evaluated in some way, and there's all sorts of different ways to do that. But we're sort of lucky in Hawaii that there's been a previous modeling effort done, these range envelopes, that we're able to use to compare with our models so that we can use them as a validation. And I'm going to be talking about that a little bit. So this is the species distribution or habitat suitability model for the Cyanea kunthiana. And the darker red means it's more suitable. This is a comparison with the other modeling approach, the range envelope. And you can see the idea here is we want to compare the two and see how well they jive, how close are they. And here we have a lot of confidence that the models are working very well. Both models are developed using different data. So they're both showing a very similar pattern, so we have some confidence in this. You may be asking, well, if you already have a model, why are you developing more models? And the reason is that the previous range envelope estimates are really just binary estimates. So it's either in the range or out of the range. And what we're really interested in, the suitability varies across the landscape. And that's what we're really interested in, because what we want to do is use that information to figure out where our recovery populations are located. Which brings me to the third part, which is basically building those populations on the landscape. So the suitability model, as I mentioned, it has the suitability values vary across the landscape. And it's basically, the model comes out as a raster. A raster is like a picture, so it has cells or pixels, and each of those pixels or cells has a value. And in addition to the value, so that's the suitability value, and in addition to the value, it also has a space, an area requirement, so it's a certain size. And what we want to do is try to capture that information, use that information along with the population criteria that we did earlier, to basically use that to build our populations based on our criteria on the landscape, and that's what we're trying to do. So in order to do this, we don't want to do it by hand. So once again, we're using R, we're having the computer do this for us. And the basic idea is you develop some sort of algorithm so that it builds a population on the landscape. Now there's all sorts of different ways we can do it, but we decided to take a very simple approach. And basically what we do is we set it up so that the computer just looks at certain criteria and then builds the landscape. Builds the population. And so what we decided to focus on was, well, we'd like a population where the area has the highest suitability, and then ideally we'd like the population to occur where there's known records of the species. And so that's basically the way we set it up on the computer. So what it starts with is there's that olive cell there with the 0.8 and the dot in it. That's where the population starts. The computer picks that out. It adds all the cells around it that's part of the population. And then it goes through and looks at all the neighboring cells and then looks for the next highest suitability value and adds that to the population. And then it adds all its neighbors, and it does this over and over again until it reaches the size of the population we're shooting for. So once again, the size of the population is we want 400 individuals. We have a density estimate, so that translates into a space requirement. And so we're able to do this over and over again for as many populations as we want and apply that across the landscape, and that's what we're doing here. So we have all sorts of different populations. Now the challenge here is, well, which ones do we really want? I mean, there are all sorts of different ones here, and they have different values. And so this is where we come into the ranking criteria. And there's all sorts of different criteria we could use. The ones we've been focusing on are just sort of overall suitability values for the population, how many species records are in there. But there's all sorts of things we could use. And one of the interesting things, I think, about this process is that if you imagine doing this for every species that you have, building all these populations, is that you can actually look at the relationship between all those populations and their suitability values and actually focus your efforts on certain populations that have high overlap with other species, which basically means you can get more bang for your buck. You can focus on areas where the greatest conservation value is. And now, so this is the results for this species, Cyanea kuntiana. And if you remember earlier on, I gave you some of the criteria for this species. We needed 10 populations of so many individuals, and they needed to be 1 kilometer apart. Well, this is basically a visual representation of that. These are 10 populations, all in orange. They're not all 1 kilometer apart. There wasn't enough space for that, but they're all the correct size. They're all, in theory, large enough to support 400 individuals. And so this kind of gives us a way to visualize that process. And the interesting thing about it is that, often when you write criteria and you say, I want this and this and that, it's hard to visualize what that actually means in reality. And this actually provides a method for doing that. So you can actually decide, well, is that really what I had in mind when I came up with those criteria? And so the other part of the process is that once we've identified these populations, we don't actually say, okay, we're going to put a fence around that and that's what we're going to manage. These are computer-derived populations, so they're going to have weird, funny shapes and things like that. The idea is to say, well, this is what we're focusing on. This is the area we're focusing on here, and then you go back and then you use things like administrative boundaries, property boundaries, natural features, and that's how you actually draw your unit. But this helps focus that process. So what I wanted to do is talk about a couple examples of how this has been applied to hopefully highlight its usefulness. The main one is actually with the island of Lanai. It's an island in Hawaii, and there's basically one major landowner who owns most of the island. And we had proposed critical habitat on that island, and they were interested in having their lands excluded. So they had a conservation agreement. And what we wanted to do was basically evaluate that conservation agreement. Did they protect enough area for all our species? Was it in an area that we wanted it in? And so in order to evaluate that, we basically did the process I just described. We built populations across the island. We also built populations specifically within the areas that they wanted to conserve. And then we can compare and contrast. And then by doing that, we can also lay out the process. So when we met with them, we talked with them, we explained the process that we did, and then we were able to say, well, we needed this or that, and we were able to explain why we came to that conclusion. And so it helped us to improve that conservation agreement. The other example here is with the Mariana Islands, and this is more to highlight the ability for it to be applied for multiple taxonomic groups. Here we're trying to apply it across 15 islands for all sorts of different species, from bats to birds to snails. And so once again, we applied that same sort of process there. And the little figure here on here we go, down here in the bottom, that actually shows you each of the populations for each of the species. And you can actually sort of imagine, you can actually kind of envision where your boundaries of your unit would be. And so we use that in the process of helping to delineate that. So it worked quite well for that. This process is, an iterative process. We're always working on it. It never seems to end. And some of the things we're working on, one of them is a density estimate. That really drives a lot of it, so we want to make sure we improve that. We want to make sure, all the species experts are on board with our models. And then the other thing we want to try to do is apply it into future climate scenarios. Right now we're working on finishing it up within the current climate, working out some of the kinks, but then we want to apply it into the future climate so that we can look at how populations might be affected in the future. Thanks. And then finally, the one thing we want to do is apply it to multiple taxonomic groups. And the one species I want to highlight here, there's been a lot of work on the Hawaii forest birds, some recent publications, and what we want to do is apply it to that, and then also use it with our plants so that we can look at how those are related and then have more of a unified approach for the islands. And then I just want to acknowledge all the various agencies and everyone's assisted with it. Thank you guys for your time, and then if there's any time for questions, take them.
So we have time for questions. Are there any here in the room?
Hey, back to you.
So one quick question I had, what climate models are you thinking about using?
Unfortunately, we only have, I think it's, I'm sorry. Is it A1B? Is that what we're?
We have A1B, the 4.5 and 8.5.
Yeah. So yeah, so we have A1B, what was it again, Ann?
4.5 and 8.
Yeah, A1B 4.5 and 8.5. Those are the current ones we have. They have to be downscaled for the Hawaiian Islands, and so it's been a bit challenging to do that, so we only have limited ones that we can use. So.